Hello, my name is Dan Lavery. I am a senior in the Geoscience Department and in the Honors College. I worked under Dr. Moira Ridley, also of the Department of Geosciences, to calculate a solubility product for the acid-sulfate mineral jarosite. Jarosite, the mineral jarosite, which has its formula expressed on the screen, is a sulfate mineral often found in environmentally hazardous environments, such as acid mine drainages. As such, quantifying jarosite solubility will allow geochemists and environmental scientists to better understand the impact of jarosite on the environment. In addition, jarosite has been found in Martian soils, so understanding jarosite solubility can help geoscientists constrain Martian surface conditions at the time of jarosite. The methods of this experiment took place over the course of 1,100 or more days, which means that it started before I enrolled at Texas Tech as a freshman. So I was not involved in the experimental side of this. I worked to analyze the data. However, the methods of this experiment, samples of jarosite, both synthetic and natural, were sealed in syringes in solutions comprised of chloride or nitrate, as well as tetramethylammonium. Here on out, I'll refer to that as TMA, and hydrochloric acid. Samples were reacted at ionic strengths, also SI, of either 0.1 or 0.3 molal, and a range of starting pH from 1.5 to 3.5. And samples were reacted at dissolved at 50 degrees Celsius in a water bath. Samples were routinely withdrawn from syringes, filtered, acidified, and then analyzed with ICP and ion chromatography. The solubility experiment yielded concentrations of ferric iron, that's iron 3+, potassium sulfate ion, and free proton, that is hydrogen, the hydrogen plus. These data were evaluated as a function of time in days and pH in order to identify differences in solubility. Any aqueous species that were thermodynamically expected to form in this solution were calculated using the Geochemist Workbench student version software, specifically the Geochemist spreadsheet and the speciate program within that software. Since no thermodynamic data for tetramethylammonium could be found in the Geochem Workbench thermodynamic datasets, or online, data was speciated using either ammonium or sodium as a stand-in for TMA. The result of this stand-in is that there are now two data sets for each original set of data, one speciated with ammonium and one speciated with sodium. Geochemist Workbench was also used to predict mineral phases that would form in equilibrium with the solution. Speciation measurements yielded activity values in moles per kilogram, activity being the effective of a solution of an ion, accounting for the ionic strength of the solution. These activity values were then used to calculate the solubility product, or KSP, of jarosite using the proposed dissolution reaction from Madden et al. Displayed on the screen, as well as the associated solubility expression, again, KSP, this being shown on the slide. So now results. This is a plot of ferric iron, again, that's iron 3+, molality of iron 3+, that's moles per kilogram on the y-axis, and time in days since the start of experiment on the x-axis. As you can see, samples 5 and 6, which have starting pH 2. These are represented by the blue and pink symbols, released iron into solution in a much different fashion than samples 1 through 4, here represented by black, red, yellow, and green symbols, which were at starting pH either 2.5 or 3. The iron concentration for samples with starting pH 2 decreased until approximately day 125, at which point they reversed course and began to increase alongside samples 1 through 4. Now this is has the same axes. This plot has the same axes as the previous plot. However, it is plotting samples 5 through 8 instead of 1 through 6. So 5 and 6 are still on the plot, except these are now represented by the black and red symbols. And samples 7 and 8, which had starting pH of 1.5, are represented by the green and yellow symbols. As you can see, sample 7 and 8 at the much lower pH, at the lower pH, the lowest possible pH of this experiment, had an even more different fashion of dissolution in that it decreased until, again, approximately about day 125, but never started increasing again. It only its rate of decrease only slowed after day 125. This table of values is just selected samples classified by their starting pH, ionic strength, and the PKSP from the last day of the experiment. Again, PKSP is the negative log 10 of the KSP, with the KSP value again displayed on the screen for viewing ease. This is a graph of PKSP on the y-axis versus time since days in the start of the experiment on the x-axis. The PKSP values start at quite different values, but then by the end of the experiment, for this set of data, it was day 924. They converge into a much smaller range. But as you can see, there are two pockets of data indicated by the red arrows. As you can see, the lower pocket is represented by samples 1, 3, and 5, represented by the green, black, and red symbols. And then there's data from samples 9, 11, and 14, represented by the symbols in blue, purple, and sky blue. Samples 1, 3, and 5 were dissolved at an ionic strength of 0.1 molal, and samples 9, 11, and 14 were dissolved at an ionic strength of 0.3 molal. So as you can see, ionic strength has a significant difference, significant impact, and has not been fully accounted for in the PKSP calculations. Now, you'll also note that sample 7, that is represented by yellow symbols, is also been dissolved at an SI of 0.1 molal, but is anomalously high relative to the other 0.1 molal ionic strength solutions. This is consistent with what we saw in figures 1 and 2, which shows different iron 3 dissolution behavior at low pHs, which means that will need to be accounted for in the future as well. This histogram compares data for samples with aqueous media of chloride and nitrate that underwent speciation calculations with ammonium as the stand-in for TMA. Note that the y-axis is the count of PKSP values, and the x-axis is PKSP, which is again the negative log 10 of the KSP, in bin sizes of 0.2 units of PKSP units. There's no perfect agreement between these values, but you can see that the data generally falls between a PKSP value of 13 and 13.6. This histogram is for synthetic samples with aqueous media of chloride or nitrate that were speciated with sodium instead of ammonium. Again, there's the same axes as the previous graph, and bin sizes, same bin sizes as well. Again, there's not the agreement is not as good with this plot, but most PKSP values still fall between about 13 and 13.8. This histogram compares PKSP values for synthetic samples, comparing whether they were speciated with sodium or ammonium. Again, that's synthetic samples in chloride media, sorry. Again, these are the same axes as the previous histogram and the same bin size as well. These data do agree quite well, and the PKSP values tend to fall between a range of 12.8 and 13.6. This histogram compares PKSP values for synthetic samples reacted in nitrate media, comparing between those speciated with sodium and ammonium, and these converge on a quite small range of PKSP values between approximately 13.2 and 13.8. This histogram is a comparison of natural samples speciated with sodium or ammonium. And as you can see, the axes are the same. However, the bin size for this has been increased to 0.3 PKSP units because with a bin size of 0.2, is difficult to see the distribution in the data. So I had to manipulate the data distribution. As you can see, most values appear to fall between a PKSP value of 11.3 and 12.5. Now, for a discussion of the data and conclusions, the agreement of the data is quite good. Ionic strength has a variable impact on jarosite dissolution, and evidently the proposed dissolution reaction from Madden did not fully account for the difference in ionic strength, or the data reduction that the data analysis that I undertook did not account for the ionic strength of the solution fully, because there's a clear difference between samples with ionic strength of 0.1 molal and 0.3 molal. PKSP values for synthetic samples generally fall between 13 and 13.6, corresponding to a KSP range between 10 to the negative 13.6 and 10 to the negative 13. Remember, PKSP is the negative log of KSP. PKSP values for natural samples generally fall between 11.3 and 12.5, corresponding to a KSP range between 10 to the negative 12.5 and 10 to the negative 11.3. Future work will need to be done to determine statistical outliers and develop a more precise KSP value. Additionally, I'll need to explain the difference in solubility at low pH that we saw with the iron dissolution and the low pH anomalously high PKSP values. Some conclusions. Natural samples are apparently about 1 order of magnitude more soluble than synthetic samples. I'll need to do more research to determine whether or not this is a realistic figure or whether there's an error in my data calculations. I would like to thank the Honors College Undergraduate Research Scholars Program, supported by the C.H. and Helen Jones Foundation, for funding me for this project. These are works that I cited during my presentation. Thank you.